Hi, I'm Tom Schreiber.
And I'm Kelsie Ramirez.
And for our presentation, we're going to discuss and analyze an article published in the American Journal of Epidemiology titled Smoking, Alcohol Consumption, and the Risk of Amyotrophic Lateral Sclerosis, a Population-Based Study. This study was performed by a group of scientists in the Netherlands. According to ALSA.org, ALS was first found in 1869 by French neurologist Jean-Martin Charcot, but it wasn't until 1939 that Lou Gehrig brought national and international attention to the disease. Amyotrophic lateral sclerosis, or ALS, is a progressive neurodegenerative disease that affects nerve cells in the brain and the spinal cord. Motor neurons reach from the brain to the spinal cord, and from the spinal cord to the muscles throughout the body. The progressive degeneration of the motor neurons in ALS eventually leads to their death. When the motor neurons die, the ability of the brain to initiate and control muscle movement is lost. With voluntary muscle action progressively affected, patients in the latter stages of the disease may become totally paralyzed. 50% of patients die within 3 years after onset of symptoms, mainly due to respiratory failure. Motor neuron degeneration in sporadic ALS is considered to be a multifactorial process consisting of both genetic and environmental factors. Smoking has been a possible risk factor for developing the disease ALS. The research study was performed in the Netherlands between 2006 among 494 patients and including 1,599 controls. Several studies have investigated environmental risk factors, but only smoking has consistently been posited as a possible risk factor. Cigarette smoke could increase the risk of developing ALS through several mechanisms, including inflammation, oxidative stress, and neurotoxicity caused by heavy metals and other chemical compounds present in cigarette smoke.
Okay, now I'm going to be speaking about the connection. The aim of our study is to prove the relationship between alcohol and or smoking ALS. Previous studies and 2 recent reviews have investigated the relation between smoking as a risk factor and ALS based on the best available research, with the authors concluding that smoking could be a risk factor for ALS. In these studies, there were several methodical drawbacks that had a hampering effect on the evidence. These drawbacks included, but were not limited to, small or selected study samples, the use of death certificate data, and insufficient account of potentially confounding factors such as educational level and alcohol consumption. With these limitations in mind, the goal is to have accurate evidence for the study. The aim of our study was to provide Class 1 evidence for a possible relation between smoking and or alcohol consumption and ALS in a large, representative, prospectively recruited incident patient group in comparison with age, sex, and geographically matched population-based controls. This prospective population-based case control study in the Netherlands provided evidence that cigarette smoking is independently associated with an increased risk of ALS and that alcohol consumption is independently associated with a reduced risk of ALS. We expected alcohol consumption to be a confounder of smoking, but it appeared to be associated with a reduced risk of ALS independently, and no significant interaction between alcohol consumption and cigarette smoking was found in our study.
For the techniques used in this study, the research study is a population-based quantitative correlational research study. According to the article, from January 1st, 2006 to June 30th, 2009, we performed a population-based study, prospective ALS study of the Netherlands, aiming at complete ascertainment of all patients with ALS in the Netherlands. Patients with ALS were recruited through multiple sources: neurologists, rehabilitation programs, patient support associations, and a website. The Netherlands provides healthcare to the public and is composed of a substantial infrastructure. Both of these things make for a great climate of a population-based research quantitative correlational research study. For the techniques used in the study, patients were given questionnaires about their lifestyle. Data on cigarette smoking, highest level of education, and alcohol consumption were recorded by questionnaire. This questionnaire was a modified version that was used in a previous study on the relation of smoking to education and occupation. Detailed data were collected on the age at the start and cessation of smoking and alcohol consumption, as well as the daily number of cigarettes smoked and units of alcohol consumed. Smoking and alcohol consumption status were categorized as never, former, or current at the time of disease onset, so before the diagnosis. Current smoking or current alcohol consumption was defined as smoking or drinking at the time of onset of weakness or swallowing and speech difficulties. Lifetime cigarette smoking was expressed in pack per years. This was calculated by multiplying the number of packs of cigarettes by the years spent smoking. Lifetime consumption of alcohol was expressed as the total number of units of alcohol consumed. All questionnaires were coded prior to the processing and analysis to ensure blinding. Response rates were recorded for both patients and controls. Additionally, the persons gathering the data were blinded with regard to the hypothesis being tested. If data were found to be missing or inconsistent in the submitted questionnaires, patients and controls were contacted by telephone to complete the information or correct the inconsistencies. Data entry was automated by importing corrected questionnaires into the database using a scanner. Then, for the results, this prospective population-based case control study in the Netherlands provided evidence that cigarette smoking is independently associated with an increased risk of ALS and that alcohol consumption is independently associated with a reduced risk of ALS. We expected alcohol consumption to be a confounder of smoking, but it appeared to be associated with a reduced risk of ALS independently. Current smoking also is associated with a worse prognosis.
We chose this particular article to research because we both know a lot of people close to us that smoke. Some interesting facts about cigarette smoking is that it causes more than 480,000 deaths each year in the United States. This is about 1 in 5 deaths. Smoking causes more deaths each year than HIV, illegal drug use, alcohol use, motor vehicle injuries, and firearm-related incidents. More than 10 times as many U.S. citizens have died prematurely from cigarette smoking than have died in all the wars fought by the United States during its history. Smoking causes about 90% of all lung cancer deaths in men and women. More women die from lung cancer each year than for breast cancer. The risk of dying from cigarette smoking has increased over the last 50 years in men and women in the United States. If nobody smoked, 1 of every 3 cancer deaths in the United States would not happen. This article is very informative, and we are not surprised to find the relationship between cigarette smoking and the risk of ALS. This research only continues to prove the devastating effects of cigarette smoking among the population. To the contrary, we were surprised to read that alcohol consumption actually had no major effect on increasing risks for developing ALS and that it actually has a reduced risk.
The synopsis of our article, the article highlighted cigarette smoking and alcohol consumption as possible risk factors associated with ALS. The authors hypothesized that both increased the risk of ALS in the population. The findings showed that they were correct that cigarette smoking is a contributing risk factor for ALS. The study surprisingly found that alcohol consumption was independently associated with reduced risk of ALS.
A limitation of the study was that they could not separate analysis from men and women due to the reduction of sample size. Another limitation may be that persons participating in case control studies are healthier than the general population, known as healthy worker effect, explaining the lower percentage of current smokers in the control group. However, it is not likely that our control group was healthier than the patient group, since alcohol consumption, also considered a bad habit, was overrepresented in controls. The strength of the study was that it filled the criteria for Class 1 evidence. This research adds to the current research on smoking and how detrimental it is to our bodies and health. In oncological studies, progress has been made to identify susceptibility genes which, combined with smoking, have a multiplier effect. In the future, there is a real possibility of identifying more risk factors for ALS, which will help to create a more healthy population of people avoid such factors.
Thank you.